So we'll continue with our presentations from the partner organizations. And again, we have a mixture of people who kindly made it here and some who unfortunately couldn't and who are doing their presentations online. So we're going to start with Sarah Muturu from Biovax in Kenya. And again, I'd like to ask you to stick to time, all of you, if you can. Thank you. Sarah.
Good afternoon, everyone. Thank you very much to all the organizers, everyone who made this happen, and for sponsoring the event. I'm really glad that I'm here. This is my first mRNA technology transfer program event. So I'm really privileged and I've really gained a lot of insights from the morning presentations. My name is Sarah Muteru. I'm a pharmacist. I'm the General Manager, Regulatory Affairs at the Kenya Biovax Institute. I know we have Biovac here. In fact, they made a mistake and on my tag it reads Biovac. But we really look up to Biovac because we are also a government state-owned organization. So we have a lot to learn from Biovac. Next slide, please. My presentation really will be about what Biovax is all about. We'll talk about the workforce, the facility, and what we've been able to do. My colleague, Dr. Cecilia Wanjala, I think she's somewhere in the audience. Yeah, they're right at the back. I think this is her second meeting. And for those of you who have been here before, most of this will be very familiar. And being a government institution, we sort of might look like we are moving at some snail speed, but we are really making progress. So some of the photos will just be almost the same as what was presented last year. So you'll just bear with me. And then, of course, the big question, we were discussing this over lunch with Heather and with Eva, the timeline. Everyone asks, when are you going to manufacture your first vaccine? And that's really the million-dollar question, but we are going to get there. Next slide, please. So, like I've mentioned, the Kenya Biovax Institute was established in 2021 as a company, a state-owned company limited. So it's fully government-owned, 100%. That doesn't mean that that is how it's going to always be. Because depending on the government of the day, we have seen institutions like ours sort of morph into public-private partnerships, but at the moment it's 100% government-owned, with the sole member being the Cabinet Secretary Treasury. For those of you who are government, you understand what this means. We also have a board of directors. As a matter of fact, our inaugural board just came to an end, a 3-year term board. And so we have a new board of management and or board of directors. And so we are going to start all over again to induct a new board, and we pray that they are going to share in the same vision. Next. Sorry, I forgot the gadget. So sorry about that. So we are really proud that His Excellency, the President, William Ruto, was voted by the African Union as a champion for local manufacturing. So political goodwill is really important, especially for a government institution like ours. So if Biovax fails, he will fail. And so I know that he would really want for this project to succeed. So one of the reasons why we were established is to ensure that we have the universal health coverage agenda, and not only for vaccines, but also for other health products and technologies. So in Kenya, we have about 45 local manufacturers of other pharmaceutical products. And so as a president, he's really keen to ensure that as a country, we have equitable access to health products and technologies. And then, of course, we have to strengthen, of course, the manufacturing sector and to be ready for the Gavi exit We know that most of our African countries need to be ready and to have availability of these vaccines by the time Gavi exits. For our country, it's set to be sometime between 2028 and 2029. And then sorry about that. So, of course, we need to leverage on the existing potential local skills. Of course, for vaccine manufacturing, we have to ensure that the local capacity is built and we have to train our staff. Vaccine manufacturing is still new in our country. And so at the moment, we are still trying to upskill our staff actually from the university level going forward to whoever is employed by BioVax. So this is our mandate. Our mandate is to manufacture human vaccines, biotherapeutics, including insulins, antivenoms, monoclonal antibodies, and gene therapies. I think even just from our discussion still during lunchtime, we realized that monoclonal antibodies which are used even for treatment of cancer, like breast cancer, are still very expensive and not accessible to our people. So we hope that as we continue discussing the manufacturing of vaccines, this is an area also we will start to think about and to have resources pulled towards this so that you have to start at some point and at some level so that by the time these therapies are available for other countries, they can also be available for our people. And breast cancer is still prevalent in our country, and we have to ensure that they also have the same kind of access to healthcare as other countries. The other mandate is to support technology transfer, and that's the reason why we are here, and to commercialize the research outputs from our local universities. KEMRI is known, our medical research institute is known globally, and Dr. Wanjala has worked there. And there are several institutions that have worked and done clinical trials with our KEMRI, and so this is also an area that we want to get into. This is our vision, to be a world free of vaccine preventable diseases. As much as, yes, we are talking about monoclonal antibodies and other therapies, really the main mandate, learning from our lessons post-COVID, we need to be ready with our vaccines so that we can have vaccine preventable diseases. But then, of course, our mission, I've already mentioned, is to manufacture and commercialize safe, effective, and quality vaccines, including other health products and technologies. And here, the main reason why it's a government institution is so that it's affordable. We provide quality, affordable health products and technologies. This is our strategy house. We call it the strategy house. We have the 5 pillars. My colleague, Dr. Wanjala Cecilia, actually designed this, and I really need to commend her for this. So our first pillar is commercialization. I don't want to belabor on that, I've mentioned. Then, of course, manufacturing. Then we have regulation and compliance. Eva in the morning mentioned that vaccines are meant for a healthy population. So healthy children, as a matter of fact, especially when we are talking about traditional vaccines. So regulation, ensuring that we have quality, efficacious and safe vaccines is going to be very, very key. And that's the reason why we're talking about WHO maturity level 3 and WHO pre-qualification of vaccines. So regulation is going to be key. Then collaboration, that's why we are here, collaboration. And that's how we need also to see how we can pull resources together, have partnerships with already established entities. We don't want to reinvent the wheel. And then we have the R&D. With vaccines, this is a continuous, and also pharmaceuticals, you have to continue with the research and development agenda as well. Then we have, of course, the foundation of the people culture and capacity development. The reason why we are in this business, as much as it's going to be very painful in terms of burning the midnight oil, studying, is so that future generations can look back and see that we tried and we did something, especially now that we know as Africa, we need to be ready to manufacture our own vaccines for our own African people. This slide is very important. It looks simple, but this is what we have been doing for the last 3 years. Just the fact that we have now technical staff for a government institution that 3 years ago did not exist, having the company registered, maybe for some of us might not understand, but it takes about 6 months to have the company registered. Then after that, developing the human resource instruments so that you can now advertise. It's a very protracted process when you're talking about government recruitment as a government institution. It's very different from the private sector because you have to engage different stakeholders in government. You have to ensure that you have all the regulations government has set have to be followed. And also because you're also recruiting from a very limited availability of qualified personnel. So it's not an easy process to be able to attract the right personnel. So right now, our staff establishment is 126. Out of this, we've been able to recruit 29. 9 are technical staff, and then the support staff are 20. You might want to ask why there's that kind of imbalance, but you cannot recruit without the human resource department. You cannot pay your staff without the finance department. You cannot procure a desk in government without the procurement department. And of course, the IT staff. That's the reason why it looks like this. So we are still in the recruitment process. It's not something that can happen overnight. So this is a big deal for us. Initially, when the government decided to get into this project, there were 2 old warehouses that were donated and they were run down. So what the Kenya Biovax has done is to just clean it up and do the painting, do the floors. And right now, I don't have the photos with me, but we have what we are calling the enabling works. The enabling works are setting up the water treatment plant, which is somewhere at the back of this warehouse, and also the biokill. And the reason for this is because we want to get ready to get the modules for production, which we are getting through the World Bank funding, which I will mention in my other slide. But we are recipients of some funds through the Health Emergency Resilience Preparedness Program. And so we will get about $120 million for the production equipment. So this is just in preparation for that. We hope that this is how it's going to look. It's a fill and finish facility. There are already the concept designs have been approved by our regulator. We have engaged with a consultant from India, and he has worked with Serum Institute. So already we have designs approved, and we are hoping that the moment the funds are disbursed by the World Bank, we'll be able to get the equipment and hopefully start production soon. So this is our timeline for vaccines production in Kenya. I will just go straight to the 2022 April, when the government of Kenya was appointed as the Kenya recipient for the WHO mRNA vaccine technology transfer program. And like I mentioned, in 2022 also, we started the discussions about the World Bank funding, and last year in 2023, the World Bank approved the project, and it was launched this year in May. So we are still waiting for the disbursement, because at this point I need to mention that one of the disbursement conditions was Kenya attaining maturity level 3. The regulator attaining WHO maturity level 3. So this is not within our control, but the government is doing everything it can to ensure that the Pharmacy and Poisons Board, our regulator, attains maturity level 3. We are hoping that it will be announced in the next WHA. When the WHO inspectors were here, they said that we've made significant progress, and there's a likelihood that we will be announced as maturity level 3. Yeah, so this slide is sort of, we don't have, right now, we can't say that we have manufactured. Yeah, we haven't manufactured, but we are not giving up. We know that we're in this for the long haul. And so this is just an African proverb. When a needle falls into a deep well, many people will look into the well, but few will be ready to go down after it. Most of the vaccine manufacturers who are already in this business, they'll tell you they've been doing this for 50, 100 years. I'm sure they had difficulties in the beginning. We are not going to give up. My colleague there has bags of optimism. We don't give up. Maybe because it's supposed to be an implementation session, we're supposed to show progress. But for us, this is really progress, and we have spent sleepless nights even just coming up with a strategic plan and all that. So we are not going to give up. We will dig and look into that well until we get the needle. Thank you very much. Really appreciate it. Yeah. I will take any questions, if any.
Can we do it at the end?
Yes, thank you. Thank you very much.
I'd just like to. Yes, thank you very much indeed, Sarah. That's delightful that you're ready to go down the well. And but I also just wanted to emphasize something else that you said about the condition with your World Bank loan requiring essentially the investment to take your national regulator to ML3. And I think that's been one of the features of this program. It has really encouraged governments to invest in their national regulatory authority. So that's a kind of, added benefit of this whole program. And so I hope that the funders in particular will look at that and think, yes, no, it's not just about this program. There are lots of ancillary benefits attached to it. So with that, we'll move on to the Institut Pasteur de Tunis. And Makram Asafi is online. So over to you, Makram.
Thank you. I would like first to thank the organizer of this meeting and send our warm greetings and salutation from Tunis to all the attendees, and especially to our colleagues from South African hubs, Afrigen, SAMRC, and BioVac. So we are really glad to be part of this international initiative led by WHO and MPP that witness major change, at least in my view, in a way to tackle the infection disease in low and middle income countries by not giving the access to drugs only, that means to drugs, access to drugs and vaccine, but also give the opportunities to get access to the technology that made these products. So I'm Akram Sefi, donc I am a researcher, background in immunology and molecular biology, and today I'm talking as the coordinator of our steering committee that is in charge of the implementation of this new facility, mRNA R&D facility that will be dedicated to the production of mRNA-based vaccine in the context of this nice WHO MPP mRNA tech transfer program. Next slide, please. So all started in April 2021 when Tunisia, through IPT, sent the letter of intent to be part of this initiative. And next, please, if you can, there is some animation. Yeah, okay, click. Yeah, thanks. 1 year later, Tunisia was designated as a partner in this program with Korea as the WHO training hub for biomanufacturing. And in April 2023, 4 scientists from IPT attended this kind of kickoff meetings called face-to-face meeting. Right after the designation of Tunisia as a partner, so we made this mRNA task force at IPT that it's composed of several competences. People have the background in immunology, microbiology, also bioprocess engineer and scientist, and we have also the technical transfer and IP expert. And after a long discussion inside this group and discussion with our partners in WHO, we decided to choose the strategy for IPT. Next, please. So this slide summarizes the strategy of IPT related to the mRNA program. So at this stage, we are working to implement this R&D unit dedicated to the design and development of mRNA vaccines. So this is technology. That means this is kind of short-term strategy based on technology that we will receive from Afrigen. We already received several SOPs, and we even started doing some experiments at the low scales. In the meantime, we have received this variable donation from WHO, which consists of the Intensify machine from Quantum. It's kind of automatic system able to generate capped naked mRNA. I have to mention here that human resources also will be reinforced by European Commission capacity building project. It's called Vision, that is coordinated by IPT, and also, of course, we will profit from the training hub in Korea. But we also aim at the midterm to evolve to GMP production step of mRNA vaccines. Our aim now, we are working to construct a new facility, new GMP facility, that will be dedicated to the current products that we are producing, like BCG and antivenom sera, and a line that will be dedicated to the mRNA vaccine inside this facility will be also built inside this facility. So we already identified the needed land for such facility and working with our government and other stakeholders to implement this project. Next slide, please. So to go further, that means to work on this implementation of the units. I think a short group from IPT. These are several colleagues. They have their own projects and work program, but they are dedicating part of their time to help the implementation of this unit. Hopefully, we got some funds also to hire 4 people, the project manager and the scientists, and 2 technicians to work exclusively for the implementation of this unit. Next slide, please. So here our work progress. So in August 29, we received an MPP delegate. And during 3 days, we had several meetings with MPP expert to assess the preparedness of Pasteur Institute to receive this technology. And after 3 days, this ended up by the co-signature of a report that was pretty positive for the preparedness for the reception of this technology. In March 2024, we received the Intensify machine. And 2 engineers came from Quantum to perform what's called the site acceptance test. In August 2024, we completed the work after several meetings, tens of meetings with MPP. We completed the final, that means draft for the agreement. That means the modified agreement and the work plan, and it was signed by all the parties. And in September, another team from Quantum, they came to Tunis, 4 people from Quantum, and to perform some engineering run on the machine. And we did some experiment using the machine that were pretty successful. And this October, we received the first part of the fund, 60% of fund, and we start ordering all the equipment and reagent needed for the unit. Next one, please. So this table summarizes the different training and courses received by about more than 30 colleagues in the frame of the mRNA Technology Transfer Program. So colleagues followed several online courses, and some of them were able to travel to different countries such as Italy, Netherlands, Korea, where training and workshops on bioproduction and mRNA technology have been organized. And as I said earlier, there's more training and courses will be performed in the that's mean based on the European Commission vision that is really helpful for us to for the human resources. Next one, please. So here the location of our future R&D unit. So in the backyard of the Pasteur Institute, we have a pretty new building that is the P3 lab. And hopefully in this lab, next, please. We have the second floor that was dedicated to be an office area. But during the visit of the MPP delegates, and we decided to transform this area to the future R&D unit, mRNA R&D unit. The next, please. So this is we took some photo here. So hopefully we got some funds right now. And this unit will be transformed to GMP production. This kind of pilot GMP unit. That means that it will get the HVAC system or they need equipments, but maybe we should get more funds to make it really functional as a GMP, because we don't have right now the funding to get the GMP qualified equipments. Next, please. But hopefully for the starting, we have what we call a starting lab. It's a 50-meter square located at our research development. In this lab, we put an intensifier machine, and we are performing right now either the experiment on the intensifier and also the small scales experiment based on the SOPs from Afrigen. Next, please. So here are some current projects on the mRNA vaccines development that are in progress in IPT. We have good collaboration with Moderna and to work to develop vaccines on Leishmania and rabies also. And my team also is working with, and my colleague Shauki, we are developing TB vaccines based on the constructs from Afrigen. And this will be done with UCT, in collaboration with UCT. Next, please. More projects also, they will come next. Just, yeah, some animation. Thank you. Sorry. So more projects, they definitely they will come through the partnership and collaboration we engaged in. Actually, most recently during the Pasteur Network annual meeting, our institution signed a MOU with Pasteur Dakar, Korea, Paris, and Fiocruz to work on the development of mRNA vaccines for different candidates and for, of course, for the pathogen that we are interested in. We also signed, next, sorry for the animation, yeah. We also signed the MOU with the University of Pennsylvania, Dr. Drew's lab, for a joint application to mark Arthur Foundation grant, and this to work on pathogen that really of interest to low and middle income countries. Next, and most recently, next again, sorry. Most recently, that means September, with my colleague Thuraya, we attended the Pan American Health Organization meeting, and we agreed with our colleagues in Fiocruz to work on to develop mRNA vaccine for leishmaniasis. Next. So this to summarize, that means the project is the core of the project will be, that means the mRNA technology transfer. We have what we call it project 2, the acquisition of the intensify machine, the platform R&D. This is kind of the same project. We have this capacity building project that is really very important for us, for the human resources. And we are really working and continuous discussion with our government to implement this new facility for the GMP production for our current products, but also for the mRNA vaccines. Next. So finally, I would like to thank all the stakeholders. That's mainly WHO and PP, and also with special thanks to our former director, Professor Luzir, who initiated this initiative, and to all the steering committee members at the Pasteur Institute. And next, thank you very much for it. That's it.
Thanks very much indeed, Makram. And we'll go straight on now to Khadija Bar from the Institut Pasteur of Dakar. Thanks so much. I wasn't sure that you were going to be able to make it, so great that you're here.
Thank you. Good morning, everyone. It's not quite the morning, but I wish I was here earlier. So my name is Khadija Ba. I represent the Vaccine Research Center for Institut Pasteur de Dakar. I'm very happy to be here today and to meet all of these partners, especially MPP. I believe it is the first time that I meet some people here. So I will talk a little bit about the mRNA R&D program at Institut Pasteur de Dakar. And before doing so, I would like to talk a little bit about Institut Pasteur de Dakar, a little bit of the history and how we got here. So it was established in 1924. If we count well, we are at 100 years of existence, and we are celebrating this year our centenary. So it was created to accelerate equitable, affordable, and sustainable access to health in Africa. And some of the marking events were the first convention in Senegal, the yellow fever vaccine WHO pre-qualification, because it was really a marking event. And also in 2010, the creation of Foundation Institut Pasteur de Dakar. The difference was that it was previously under the governance of Institut Pasteur Paris, and it later became regulated and under the governance of the state of Senegal. So later on, we created a diagnostics unit, Diatropix, Madiba, which is the project to commercialize vaccines in Africa in 2021. AVBL, the Advanced Vaccine Bioprocessing Lab, and in 2024, the department I'm representing, the Vaccine Research Center. So what is really the program about? What is AVBL about? I'm sorry, it seems like this is not really the right order. So yeah. So as we said, IPD is a regional public health hub, and the focus of IPD is really 5 strategic pillars. The first one is education and talent development, because we believe that training the workforce, training our researchers to develop the best capabilities is the first thing we should focus on. We have a biomanufacturing training hub right now, that offers a few mock courses, short courses as well. In terms of healthcare solutions, we have clinical laboratory services that are one of the main providers in the country, especially in terms of epidemic outbreaks. In terms of pandemic and epidemic intelligence, we have a department of public health, and the Department of Public Health really does many activities. Mainly the one is surveillance for outbreaks, epidemics, and that one is a very important one. And for VRC, the Vaccine Research Center, it sits under the biomedical research and innovation pillar and the biomanufacturing. We are a little bit in between the two, because the first goal is really to advance the research in the field and move to production. So about the Vaccine Research Center, as I mentioned it, at first it was the Advanced Vaccine Bioprocessing Lab, and later on we found out that it was important to establish a computational vaccine design unit to really process the data that is collected and help us also make much more efficient progress in the research. And we also go from preclinical to bioinformatic research. So that is what led us to the Vaccine Research Center today. So in terms of the vision of the lab, we thought that it was very difficult to go from a disruptive innovation to implementing to large-scale GMP, because it would have been time-consuming, resource also, due to the resource constraints, it would have been much more, it would have been much harder. So that is how the idea of creating the Advanced Vaccine Bioprocessing Lab came. And the idea really with that lab is to de-risk initial technology transfer, as in we are mitigating the risks, capture the know-how, so the know-how stays within IPD and it can later be shared with other institutions who need to have access to it, enable continuous improvement, and we also provide troubleshooting capabilities for manufacturing. So all of the knowledge really and the research that is done goes directly to the GMP facility, Madiba, towards the end. So it is at home. The idea is really to have our at-home lab technology and not to have to ask other institutions. So innovation with its defined boundaries, we understood that the idea and this vision took some time, and it also needed financial resources, operational resources. It needed to be scalable, but also the regulatory aspect is very important. So that is why we focused on these areas first, on collecting these means and capabilities in order to provide a good environment for the people, establish processes, have a good platform that would lead us to have the projects that we want to create. So from R&D to local manufacturing, as of now, we are mainly focusing in R&D. The goal, obviously, is to move to release and commercialization. So within IPD, we are thinking about implementing and having a DAVAX GMP pilot scale. So it's a biotechnology unit that would help us go to that would help us do the clinical trials, and after that go to Madiba, move to Madiba, which is the manufacturing facility. So in terms of the mRNA program timeline, it all really started at the training at Afrigen last year, so in April. And then in February, started with the lab refurbishment that was needed in order to begin the activities. And some purchases were made thanks to the WHO kickoff we had, and also the Intensify that was given to us. So we received the Intensify in September, and in September we started as well the installation of the equipment. That is almost completed. So what is left right now is really the procurement and delivery of materials. Also another hands-on training that is to be expected at Afrigen by the team. And we are hoping to start the implementation of the tech transfer in April. So in terms of staffing, this is the team. What we need to know is that last year at this period, the team was only made of 3 people for the mRNA program. So if we have managed to grow a program officer, myself, a project manager, scientist, we have also an engineer, quality insurance officer, technicians, bioinformaticians. So this is really what we think that the program should be made of in terms of personnel. We are hoping to grow more if we receive more resources. And obviously the focus for these people is the upskilling. We need to do more upskilling on the resources that we have. And for that, we have been able to do a couple of trainings. So we have a platform. So far, as I said, 97% of the equipment has been received. In these pictures, you see the Intensify received from Quantum. And other equipment as well for the lab. So in terms of training, we started with the first one, the mRNA technology training at Afrigen, April 3rd. And as of now, we have had numerous trainings. McGill, really many institutions have supported us, and the human and veterinary vaccinology course in October 2024. That was the latest. And these are the trainings that we plan to have, all funded by MPP. So some of them would be online and others would be on site. And that is really important for the team to undergo all of these trainings, to develop as much as capabilities as we can. So yeah, in terms of equipment, for the equipment ordered, we have 97% ordered, and for the one delivered, we are at 85%. That is because of the procurement, and that one has always been a bottleneck in our countries. It is a bit harder to really to have control on that part, but I think we are confident that we will get there and still be on time for the delivery. So this is it was a little bit to discuss the process that we have received from Quantum, given that the machines that are used as well are the Quantum's machines. So in terms of vaccines portfolio at IPD, we have established with the Ministry of Health a surveillance network that captures really all the regions across Senegal. And thanks to the reference laboratories that we have at IPD, we are able to identify strains, and with the data that is collected, we are able also to identify the best vaccine candidates. And the 3 vaccine candidates that we have selected are Rift Valley fever, Creamy Congo, and Disease X. So right now, the next focus would be the Rift Valley fever vaccine. And these are our partners. The objective is first to recruit and train the AVBL staff to develop the mRNA R&D capabilities, build an mRNA lab that is already under progress, and starting the establishment of the rapid bioinformatics units. That is the computational vaccine design unit that I mentioned. So we want to thank also all these partners who have helped establish the capacities that we have at IPD right now, and really they have supported on many aspects. We have talked about the operational resources, we have talked about the human resources as well, the processes, that has been very important. And that is it. So Jahajef means thank you in Wolof. Thanks a lot.
Thanks so much, Khadija. Now, next on the program is Nigeria, but we seem to be having some technical problems there. So we're going to go straight on to Biomanguinhos. So, Patricia Neves, over to you.
Okay, thank you. Good morning, or good afternoon for you. For us in Brazil, it's good morning yet. Okay. So my name is Patricia Neves. I'm the head of RNA products development in Biomanguinhos, and I'll talk a little bit about the RNA platform implementation and inside this program from MPP and WHO, and the progress we've made during these 3 last years. The next, please. So Biomanguinhos is one of the units of Fiocruz. Fiocruz is the major public health institution in Latin America. We are a governmental institution, a non-profit, and the Biomanguinhos mission is to develop and manufacture vaccines, biopharmaceuticals, and in vitro diagnostic kits to our Ministry of Health. As you may know, Brazil has a unified health system, so every Brazilian has the right to health, and the mission of Biomanguinhos is to lower the price to our Ministry of Health. So Biomanguinhos is, as I told you, is dedicated to the innovation production at the service of public health in Brazil. Next, please. I'll focus today in our R&D capabilities in Biomanguinhos. So we are a very particular institution because we are able to go since the applied research in collaboration with other institutes of Fiocruz through concept trial, experimental development, non-clinical trials, scaling up clinical studies, and registration, which allow us to move forward to industrial production and deliver these products to our Ministry of Health. Next, please. So related to the RNA program, now we have 28 employees directly paid with RNA funds, the financing we have already, but we have more than 100 employees in several areas that are dedicated to the RNA program, including people from QC, QA, manufacturing, regulatory aspects, and other shared resources we have in Biomanguinhos. For R&D area dedicated to the RNA, we have more than 700 square meters of area. And in terms of manufacturing, we have more than 1,800 square meters. And this area is dedicated to the plasmid production in GMP conditions, IVT, and also fill and finish. Next, please. So since we joined the program, our strategy was since the beginning to develop our own product, and our strategy was to skip everything we could related to the deposited patents. So we started working with self-amplifying constructions, but during this way, we realized that it would be very difficult to register a COVID-19 vaccine using this technology, because in Brazil, almost everybody is vaccinated against COVID. And the study we need to perform in phase 3 would be a comparative study, non-inferiority clinical study. So we started work with the conventional constructs, and we've designed our own 5-line, 3-line UTRs, and we engineered the spike protein using bioinformatics to skip the patents, and we've also selected freedom to operate ionizable lipid, and with this candidate, we moved forward with our COVID-19 vaccine. That would be the first one we plan to register in Brazil. Next, please. So other aspect that we addressed during our development was the use of modified uridines. We have chosen to work with pseudouridine, that's freedom to operate. And we got very good results in the comparison between pseudouridine and methylpseudouridine. Next, please. This is one of the results we've got. We immunized K18 mice with just one dose of our vaccine, of our candidate, and we followed the neutralization antibody responses. And what we saw is that we have much better neutralizing antibodies using pseudouridine when we compare with N1 methylpseudouridine. And we are investigating deeply this mechanism to see that if we could have better, even better memory responses related to the use of pseudouridine instead of N-methylpseudouridine in mRNA vaccines. Next, please. During this time, as I told you, we've tested more than 20 combinations of ionizable lipids and constructs, fully developed at Biomanguinhos. Next. And we've got a very good candidate that reduced the viral loads in organs and oropharynx, 100% protection in a lethal challenge using K18 mice, robust T-cell responses, and high neutralizing antibody titers when compared with the commercialized vaccine. In our case, we compare it with Pfizer's vaccine because it's the vaccine that's being used in our national immunization program. Next. These are our non-GMP facilities. So with our financing, we were able to buy the whole package of R&D equipments. We are using also the N-Intensify Mini from Quantum, also the manual tester from Quantum, and this is to bridge our R&D process with our GMP process, and also other instruments to purify the RNA to filter using this TFF machine. Next. And those are the equipments dedicated to the encapsulation. So we have the whole package from Precision Nanosystems, and also the Blaze, that's the pilot scale equipment, and also the TFF that we are using from Cytiva, and the Zetasizer to control the nanoparticles. Next, please. And this is our GMP facility. It's inside our pilot plant in Biomanguinhos. So our pilot plant is already qualified for GMP production. This is in the third floor of this building. And this is the first RNA GMP area in Latin America. And for this area, we have chosen the equipments from Quantum, the N-Intensify MIDI, and the Precision Nanosystems GMP encapsulator. Next. And we were able to manufacture our first engineering lot in this MIDI machine. Those are the results for the integrity of the RNA. The final integrity is above 80%. So it is inside our criteria. Next. This is the encapsulation test. As you can see, we got very good results with this nanoparticle. This is already including the downstream process for the nanoparticles. We are now in a 4-liter scale, which is very good, and it's enough for our clinical studies. This production will go to the toxic studies next month. And also we've tested, next, please. And also we've tested our fill and finish process, including the sterile filtration. And this lot, as I told you, we go to the toxic studies next month. Next, please. In terms of quality control tests, we have already established more than 70% of the release for the drug product. We are now waiting for the installation of our last equipments, the HPLC CAD, and also our mass spectrometry equipment to finish the tests. Next. And we've performed also preliminary stability studies, and we were able to see that our product is stable at least 60 days in 2 to 8 degrees, and also in minus 30 degrees. And now with this engineering lot produced in a scaled up process, we will perform again another preliminary stability studies to see the long-term stability of our product. Next. In terms of quality assurance, we were able to establish 100% of the documentation to transfer the process from the bench to the pilot plant. So this process we consider is totally transferred. Next. And now we are finishing our non-clinical studies with the dose spare studies. This is finishing this month. We are going to the toxic studies next month. Our GMP area for the pilot scale is ready. Our first engineering lot is in progress. We will finish by the end of November. Our phase 1 clinical development plan is ready. So we are planning to have all the data necessary to move forward to the clinical studies in the beginning of next year. And then we will wait for the comments from our regulatory agents. And we are all prepared to move forward to the clinical studies. Next. So with the skills we have today, since the early development and proof of principle of RNA products, and the skills that we are building now are related to scaling up and GMP production, we think that we are able to commercialize a product, totally new product, within the time necessary to perform the non-clinical studies and clinical studies for a new target. So I think that the time will depend a lot on the understanding of our regulatory agency related to the need to the non-clinical studies, mainly toxic studies again, and the time necessary to have the clinical data. So 3 to 5 years, depending on the understanding of our regulatory agency. Next. In Brazil, we have a very good position to be sustainable with our RNA portfolio, because Brazil has a unified health system, and our government is seeking to include our vaccines in our national immunization program. So we are moving forward with COVID-19 vaccine that's already included in our annual vaccination program. We are also moving very fast with the RNA vaccine. It's already in non-clinical studies, and also leishmaniasis vaccine and other targets that are very important to our region. We are discussing some consortium that we started to discuss during our regional meeting. My colleague, Macron, already mentioned the leishmaniasis vaccine that we are discussing with Pasteur Tunis, and also other targets, for example, tuberculosis, that we are discussing to test together our vaccine with the vaccine from South African consortium. And also we are involved in the lipids workforce and a test from MPP WHO initiative. And we are involved now also in the development of therapeutics using RNA. Next, please. In terms of investment, we are being very well financed by our local governments, since we are public governmental institution, but we were also financed by PAHO and the German bank and other private donations. And now we are supposed to be fully financed by our government to finish the COVID-19 vaccine development. But since we are having a lot of currency fluctuations, maybe we will need more support to finish to finish phase 2 and 3. And also we are seeking for new fundings to construct our commercial RNA production area. Since we are working with a lot of different products, we will need our pilot plant facility to produce the material for clinical studies. So we are planning to move forward and construct a commercial area dedicated to the commercial production of RNA products. Next. So we, as already told you, we are partnering a lot of different institutions through the MPP and WHO initiative, Afrigen, PAHO, MPP, also Synergium. And I highlighted here some partnerships related to the regional, to increase regional capacities in supply chain. So IBMP is planning to incorporate enzymes production, regional enzymes production for IVTs. And we are also talking to Seprocor and Elise to regionalize lipids production in Latin America, what I think it would be very important to the sustainability of this program. Next, please. So this is almost the whole team involved in this project, our financiers, and I would like to talk our team and mainly Professor Ana Paula Diniz Anbon, who is my partner in this program development. Thank you.
Thank you very much indeed, Patricia. And now I'd like to hand over to Herman Sanchez Alberti from Synergium. Please.
Okay, so good afternoon, good evening, and good morning for our Brazilian and Argentinian colleagues that are connected. It's a pleasure to be here with you today. So I would like to start saying congratulations to the Afrigen team. It's incredible the job that they've been doing during these 3 years. Our team is currently performing the training, and they are extremely happy with the knowledge that they have been transferred to them during these 2 weeks. So today I will take you through our journey in the mRNA technology transfer and share our future plans for implementing the technology in Argentina. Sorry, here. So it all began in 2021 when we were selected to be part of this program. 3 years has passed since that time, and remarkable process has been achieved. In March 2022, our team received the first introductory training in research and development here in Cape Town. From that time, we started working with the technology at the lab scale. In January 2023, we formalized the technology transfer agreement under the mRNA program. And by October 2023, we signed a technological cooperation agreement with regional entities such as PAHO and the Ministry of Health of Argentina to strengthen the local capacities in the region. Then, during 2024, we established the readiness work plan and began acquiring the necessary equipment for the technology transfer. As I said before, we are currently performing the training required to carry out the formal technology transfer. And looking ahead for 2025, engineering and demonstration batches will be executed, paving the way for achieving the tech transfer. I would like to mention that in July of this year, we announced our work on an H5 influenza vaccine candidate, leveraging the mRNA technology transfer information that we have received. So, our progress in the mRNA technology wouldn't be possible without the dedication and expertise of our team. So we have a small group of development and research and development team, but they are very well trained. At the heart of this project, we have the research and development team. Here we have 11 scientists, including 5 PhD, different professionals, biochemists, biotechnologists, biologists, and also university students. But we also have other areas of professionals that are actively involved. A total of 33 staff are involved in the project, with people from regulatory affairs, quality, engineering, and also the extending team from the legal department, purchase, and all the financial support. But let's dive into the current mRNA production capabilities and the infrastructure that we have in our country. Currently, we are conducting non-GMP activities from pDNA manufacturing, mRNA production, and encapsulation. And the GMP activities are planned to begin in 2 years. Simultaneously, we are building GMP capacities, targeting an annual production of up to 50 million doses using the standard mRNA manufacturing platform and also the intensified process from Quantum, which was acquired with the financial help that we received from the Pan American Health Organization and the Canada government. Currently, our current non-GMP, this is our research and development lab. So it's an open lab that is designed to tailor different specific activities. One area is dedicated to perform IBT here. Other area is for downstream purification. And we have also other area for performing all the analytics. We have made a substantial investment in the state-of-the-art equipment, funded entirely through our own resources. This includes systems such as the ACTA for purification, PNI for encapsulation, CANAWAR device also for encapsulation, and we invest a lot in acquiring analytics equipment. As we move forward, our primary focus is to transition from research and development to GMP manufacturing. And with this vision is that we have initiated the construction of a new facility. As you can see here, the mRNA building is located at the back of our campus. This is our campus. We have a current fill and finish facility that is in operation. In the last 2 years, we started building the utilities building first to supply not only the mRNA facility, but also an expansion of our filling lines. The GMP facility, so it's planned to be constructed in 2 stages for financial reasons. In the first stage, we are constructing the total shell of the building, covering 240 square meters, which include the research and development areas. This stage is already underway and is expected to be finished on Q2 2025. The second stage will focus on converting these GMP areas these research and development areas into GMP compliant spaces and building the support areas also. This transformation will equip us with the infrastructure needed to meet the regulatory standards for GMP manufacturing. It's important to highlight here that with additional financial support, we could significantly accelerate this timeline. So the GMP facility is designed to comply with the most standard practice. And as I mentioned before, it will have two mRNA platforms, one for the standard production and one for the intensified platform. As you can see here, the new building, the construction has been under construction, and we are finishing the first stage by the mid of next year. And while this project has been a total estimated budget of $20 million approximately, and a 2-year timeline. But now let's jump to the training that we have performed in this program. So the training courses that we have performed and planned for the next year have been strategically selected based on our needs. One of the main topics are here, basically from facility design, tech transfer, IP, quality, manufacture, and analytical courses were performed and are planned. More than $200,000 is being planned to be invested in human resources. Our research and development program basically includes a diverse set of candidates. We started working, of course, with the SARS-CoV-2 as a part of the platform development activities, but from there we expanded to the influenza field, which is our main focus right now. So the first we started working with pandemic influenza with H5N1 because of we understand that this is a monovalent and it will be more easy for us to have a prototype. And we are working with linear and with circular RNA, but our last objective is to move to seasonal influenza for sustainability reasons. Looking ahead, we are also leveraging the platform for other applications. These include vaccine for rabies, hantavirus, feline leukemia for the veterinary industry, as well as other neglected tropical disease. So, well, this slide highlights our network of partners who are helping us to advance in the mRNA technology. So we have established collaboration with leading institutions such as PAHO, the Ministry of Health of Argentina, universities like the University of Buenos Aires has been very supportive on the antigen design and preclinical tests. With Annelies Malbrand, we have signed an agreement for lipid manufacturing inside the consortium. BARDA gave us a very valuable feedback on the facility design, which is key for our GMP operations. And beyond the mRNA program, of course, we maintain regularly conversations with our regulatory bodies, like ANMAT, ensure to develop not only the platform, but also to design the facility according to the requirement. So this slide represents some of the key achievements we have made in the development of the mRNA-based vaccines. Basically, we have performed 2 proof of concepts. The first one was using averaging DNA plasmid. Basically, here we evaluate various doses of mRNA. The formulation elicits a robust anti-spike neutralizing antibody with titers significantly higher than the commercial vaccine used as a reference. But then we shift to our proof of concept of the influenza, of pandemic influenza, using our own design. Here, the functional antibody response is measured, you can see here, with the agglutination inhibition test. Sorry. What is this? As you can hear, we have designed different prototypes. Both of them outperformed the control, which was a recombinant protein with adjuvant with MF59 as using as an adjuvant. So as we look to the future, sustainability is at the core of our strategy for strengthening mRNA technology. One of our immediate focus is the development of pandemic influenza vaccine. However, for our long-term strategy is to shift toward seasonal influenza. This transition is critical for sustainability, as seasonal influenza offers a consistent demand, ensuring the financial viability of our mRNA platform. So our current fill and finish operation are influenza egg-based vaccine, the trivalent, and we understand and we have the influenza market in Argentina. So our final objective is to transition from pandemic influenza to seasonal influenza and to replace our egg-based vaccine for an mRNA seasonal influenza. To achieve this goal, we need to perform the transition from research and development to full GMP manufacturing. By 2026, as you can see here, our GMP facility will be fully operational, enabling us to produce clinical and commercially batches. This will need substantial financial support. Also, other funds that we will need to be raised are for performing clinical trials, which are expected to be performed in 2028. So these are 2 areas, finishing the facility and performing clinical trials, that require the most of the investment. Also, we are looking for consultation in CNC and clinical design, clinical trial designs, and we also plan to establish Synergion as a CDMO for other mRNA products, allowing us to offer end-to-end solution from drug substance to their product, and further diversify our revenue streams. Finally, my last slide answered this critical question regarding the time to market, and we divide it into 4 different pathways depending on the stage of the product development that we receive and our role in the process. So in the first pathway, if we act only as a CMO, just for fill and finish, we could begin operations sooner. This include conducting a formal tech transfer for the drug product and obtaining the unmatched approval by 2026 or 2027 here. If you wanted to go for a full end-to-end manufacturing, including drug substance, we would need to have our drug substance GMP facility operational. So by the end of 2027, we estimated to have the NRA approval. For a product, if we receive a product that is in advanced clinical stage, and we would need to prepare clinical protocols and all the national regulatory approvals, we estimate to perform phase 2 and phase 3 by 2029 to get the commercialization approval by that time. And if we wanted to perform our own development starting from the preclinical phases, this process would take longer, as you can see here. Clinical trials from phases 1, 2, and 3 could run through 2027 to 2030. Regulatory approval will be achieved by 2031 in a very optimistic scenario. So central to all of this pathway are the readiness of our GMP facility, and also the time required to bring an mRNA product to market depends heavily on its starting point. For advanced products, it could begin as early as 2026, but for early stage, it could extend up to 2031 in a conservative approach. So with that, I will close my presentation. Thank you for your support and for your attention. It's been a pleasure to be here.
Thank you very much indeed, Herman. And finally, for this session, I'd like to turn to Darshan Parekh from Biological E in India. Darshan, the floor is yours.
Thank you. Am I audible?
You are. And we can see your slides. So thank you.
Okay, perfect. First of all, good afternoon, everyone, to the folks in South Africa, and greetings to the attendees who are spread across the globe. I am Darshan Parekh. I am working as a senior consultant for the mRNA platform at Biological E, which is based out of Hyderabad in India. And I'm presenting here in behalf of Dr. Ramesh Mathur, who is the senior VP of R&D at Biological E and currently heading the mRNA platform at Biological E. So I'm going to give you a very quick and brief overview of what our current status is in terms of the mRNA platform and what our capabilities are, along with the infrastructure and the team structure. So I'll jump straight into the slides. Next, please. So I'll start with the team structure. So Biological E started working on the mRNA platform early 2022. And there was a set process in mind. As we can see, there are these fundamental pillars that define the process of the mRNA platform. So starting from antigen design, followed by IVT. Obviously, there is an intermediate step of gene synthesis, plasmid scale-up, linearization, and purification as well, followed by the IVT, that is the drug substance, followed by LNP formulation, that's the drug product, and downstream analytics for the DS and DP, and eventually the biological evaluation. So based on these 5 fundamental pillars, we hired a team of 10 scientists that are currently working on different mRNA candidates. And we possess capabilities currently to design manually the antigen of interest, have plasmid scale-up capabilities as well. We are currently outsourcing the gene synthesis, but we plan to bring that in-house as well. And then, of course, we have the downstream capabilities of the drug substance production, formulation, and the analytical techniques. We also have access to the CRO services as needed for any biological or animal studies. However, the downstream sample processing analytics for the biological studies will be done in-house as well. Next one, please. So in terms of the infrastructure, we currently have a non-GMP lab set up for the mRNA program with stringent guidelines for under nucleus-free operation. And we do have a GMP grade facility, a pilot plant facility, which is under planning and construction, and we expect it to be completed by 2026 or early 2027. And we are having an area of approximately 400 square meters dedicated for the mRNA processes. Now, please bear in mind that it's going to be in conjunction with the other platforms that Biological E is currently pursuing as well. So we have other traditional vaccine manufacturing programs, which for the bacterial and the viral vaccines. So along with that, the mRNA platform will be attached in terms of the GMP grade facility construction. However, in the meantime, if we are able to reach to a stage where we have to perform GMP grade studies, we have existing GMP facilities that, as I mentioned, for other vaccine programs that Biological E is working on. We could very well repurpose those facilities and have GMP grade mRNA DS and DP produced in those facilities. We also have most of the equipment procured in-house that allows us for plasmid gene synthesis, plasmid scale-up, linearization, DSDP synthesis as well, and most of the analytics are also performed in-house. Next, please. So these are just representative images of some of the equipments in the lab. On the left is the ACTA, which is used for the purification of the drug substance. The middle picture is of the nanoscaler, the NOR, which is used for the formulation, LNP formulation. And on the right is a fragment analyzer that is used for DS analytics, basically for size and integrity. And of course, in addition to these, we have a slew of other analytical equipments as well to define the identity, size, sequence as well. Next, please. So in terms of the capabilities, Biological E, as I mentioned, has invested since the early 2022 in terms of creating or establishing the mRNA platform. And the efforts have been implemented in-house, independent of any external training. And the team of scientists that have been assembled to prosecute the mRNA platform in-house is based on their prior expertise with different domains that are related to the mRNA platform, such as the molecular biology, RNA biology, as well as the nucleic acid delivery. So these are a team of SMEs who have prior experience with the mRNA platform or platforms that are related to the mRNA technology. And in terms of clinical development and commercialization as well, company already has a lot of expertise to their other vaccine programs. So in case of translating from or tech transferring from R&D to GMP grade, there is existing resources within the company that will help us set up a GMP grade pilot plant with ease as well. And of course, the most important point out here being that the company is also partnered with the WHO MPP Tech Transfer Program, which I think brings in a lot of value. So because it's such a niche area, as we all understand, it cannot be conquered independently. So our association with the WHO MPP Tech Transfer Program and training to Afrigen, it adds a lot of value for us to validate our existing processes and also fill in the gaps wherever we see it's necessary. So in terms of training, our team has already completed the introductory training at Afrigen back in 2022. And we are scheduled to receive a scale-up process training next month in December 2024. So we are definitely looking forward to the scale-up training as well. Next slide. In terms of funding, Biological E has completely self-funded the entire efforts on the mRNA platform till date. And we plan to continue pursuing multiple mRNA-based vaccine therapeutic programs. We are currently pursuing some non-COVID vaccine programs in-house, and we have finished animal proof of concept studies as well. And we plan to implement some therapeutic programs as well. For confidentiality reasons, we cannot disclose the programs as of date. However, as I mentioned, the resources are very well dedicated for the pipeline generation that are based on mRNA platform. And we also have ongoing academic collaborations with labs based out of the US that have been going for over a year for various target identification as well as for licensing of novel cationic lipids for the delivery of the mRNA drug product. So at this point, of course, we are self-funding, but at the same time, we are also looking for partnerships to different organizations or collaborations to different labs that will allow us to expand our pipeline and portfolio, not only in vaccines, but to also different therapeutic programs. So I think that's it from my end. Thank you.
Great, thank you very much indeed.